On this computer. Okay, officially, let us start. Good morning. This is MED 110, Anatomy Physiology 1. This is week 4. Dr. Garais, Alexandria campus, and it is Wednesday morning. And not introduction to course. I always do that. Now, of course, what is due today? All week 3 stuff. So it is Task 3, Discussion 3, and Lesson 3 is due today. And if you haven't done it, you got till the end of closing of business. So I'll give anyone who's missing that stuff leeway. Now, go to Task 4. What are we doing this week? Well, this week, if you recall that diagram that we had before, this diagram, which is this diagram, we already talked about atoms, molecules, macromolecules. Last week is organelles, cells, and tissues. So we are now going to talk about this thing here in the cell, which is called the nucleus. And if you call the nucleus, had DNA and RNA in it. And the DNA is essentially the blueprint of who you are. And the RNA is kind of like a foreman or a messenger that will take that DNA code and then translate it onto the ribosomes, and the ribosomes will make specific proteins that will, tell your body to go do things. So today we'll be talking about, of course, the DNA is in the nucleus and its replication, protein synthesis. How do you make proteins, and we already know protein is a bunch of amino acids, but what does that mean to us, and why is that so important? And real quick about cell growth and division and how cellular differentiation. We kind of know a little bit about cellular differentiation already. Remember when we talked about the cells and the cell membrane, and it had proteins on it? That is, we already know how cells can look like other cells, look like other cells, and how cells can differentiate from each other because they have glycoprotein coats which identify themselves as either of what they are, and kind of like a little jacket. So if you wear, like, for example, when the doctor wears their jacket, you know that the doctor has a little tag and has the white coat. Well, the coat, the cellular differentiation, we already know that's also a lock and key fit, right? And that's how medications work. That's how our immune system works. So you kind of know this fourth part. So let's jump right in and let's talk about nucleus and DNA and the structure of DNA. And in order to go do that, let's go to there's a nice video in lessons. Why do I always do that? So in lesson 4, there is a very nice video. This video right here. Now, don't worry about this and this. They're nice accessory lessons, but there is, I think, we'll look at here. I think in this cell DNA, there's nice notes that will supplement here all the things that the video will be talking about. So if you're one of those people who likes to read, it's this stuff in here. Remember we talked about like DNA and how there is a specific shape to DNA, and that is the double helix. And we're going to show I'm going to show you the video, and then I'm going to stop the video and then talk in between to explain it. And remember we talked about the chromatin inside the DNA, and inside the nucleolus, it got really, really dense DNA. Well, here's a lovely illustration how exactly DNA can like coil itself and then coil upon coil and actually form even more tightly compact coils. And this is how you get all of this information, and this is all chemical information, right? This is all chemistry into these tightly wound little balls, and then it gets into chromosomes, and then each person has 46 chromosomes, 23 from mommy, 23 from daddy, and all 46 chromosomes. If you could see all this information, it has, now you could see it's kind of like infinity. It seems like infinity, but it is essentially everything of who you are is inside this DNA, and you can now understand how. Like if there's a chemical code in here, this is already complex, but then you coil it and then coil it again, and can then coil that and then pack that into chromosomes, and now you see why chromosomes are like drawn the way they are drawn, like a little X, right? Because that's what they look like in real life when they're all coiled. So remember, the nucleus has DNA. And inside the nucleus, there is loosely packed DNA, relatively loosely packed DNA. But in the nucleolus, the the deep core of the nucleus, that's where you have the really packed and tightly coiled. And all of this is because, remember, we talked about hydrogen bonds. Hydrogen bonds are very flexible, and they can collapse and then they can coil, and it's nice because then we have every single bit of information of who you are is in this setup. So these are nice notes to look at, but let's look at this video because that's like the main part. This video has like, it's a very, I usually don't show the video, have you guys do it, but this video has a lot of really nice things in it.
Okay.
So
it's just beautiful, isn't it? It's mesmerizing. It's double helix exciting. You really can tell just by looking at it how sort of important and amazing it is. It's pretty much the most complicated molecule that exists, and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. And so multifariously awesome that if you took all of it from just one of our cells and untangled it, it would be taller than me. Now consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to the sun not once, but 600 times. Mind blown yet? Hey, you want to make one? Of course, you know, I'm talking about deoxyribonucleic acid, known to its friends as DNA. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. And it does the same thing for pretty much every other living thing. I'm gonna go out on a limb here and assume that you are a human, in which case every body cell that you have, or somatic cell in you, has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. DNA is nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid, or RNA. Now, if you can make your mind do this, remember all the way back to episode 3, where we talked about all the important biological molecules, carbohydrates, lipids, and proteins. Now, ring a bell? Well, nucleic acids are the 4th major group of biological molecules, and for my money, they have the most complicated job of all. Structurally, they're polymers, which means that each one is...
all right, let's stop. Now, we already learned that like biochemistry and the biological molecules, they're made out of carbons, hydrogens, oxygens, and nitrogens. It's the very basics of who you are. And we're going to go now over what each of all these little colors mean. Now, the word polymer is, poly means many. And a polymer is just like Lego. You take little pieces of Lego and then if you string them all along, you're gonna make a big chain of something. Now, this is a theme of not only the code, but also of proteins. Protein is also a polymer, and each amino acid gets strung along, and you'll see how that shapes together. So the code is like this one big string, and then also the protein is one big string. So the DNA code, and if you could see here, all these here, these are all the hydrogen bonds. And then when they start to twist, when they start to bend, it makes that classic double helix, that winding staircase structure that he showed in the beginning of the video. So let's commence.
Up of many small repeating molecular units. In DNA, these small units are called nucleotides. Link them together and you have yourself a polynucleotide. Now, before we actually put these tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's first take a look at what makes up each nucleotide. We're going to need three things. 1, a 5-carbon sugar molecule, 2, a phosphate group, and 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like an intertwined microscopic double spiral staircase, basically just a ladder, but twisted, the famous double helix. And like any good structure, we have to have a main support. In DNA, the sugars and phosphates bond together to form twin backbones. These sugar-phosphate bonds...
All right, so let's look at this. Now, ribose is a 5-carbon sugar, and if you you don't know anything about biochemistry yet, so if you look at this, it's how many, it goes, you see how many points are here? 1, 2, 3, 4, and where this oxygen is, that's a carbon double bonded onto an oxygen. So that's ribose. That's a 5-carbon sugar. And remember, if you remember your medical terminology, anything that says OSE is a sugar. So ribose, fructose, lactose, and the most important, glucose, which is a 6-carbon sugar. Okay, so you see this, it's like a pentagon. So that's how you know, hey, that's 5 carbons. So that's got to be a 5-carbon sugar. Now, these are like rungs on the ladder. So they call this thing, when the 5-carbon sugar, right? This is part of what they call the backbone. And if you'll notice, and they're going to mention this in the video shortly, you see how it's how like the little oxygens are facing up one way, and now the little other oxygens are facing down in another way. And remember that, because that's important in a moment. All right, so this is the backbone, and then we're going to be, and then the bases, the adenine, thymine, guanine, and cytosine. And it's just easier just to remember them as A, T, G, and C. But those are nitrogen bases. And remember, we talked about acids and bases. Bases have hydroxide ion. So this particular bases have hydroxide ion with a nitrogen, hence the word nitrogenous base. All right, let's go.
On each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar phosphate backbones, you'll see that one appears to be upside down in relation to the other. One strand begins at the top with the first phosphate connected to the sugar molecule's 5th carbon, and then ending where the next phosphate would go with a free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'. I've always thought the deoxyribose with an arrow, with the oxygen as a point. It always points from 3' to 5'. Now on the other strand, it's exactly the opposite. It begins up top with a free end at the sugar's 3rd carbon, and then the phosphates connect to the sugar's 5th carbons all the way down. And it ends at the bottom with the phosphate, and you've probably figured this out already, but this is called the 3' to 5' direction. Now it is time to make ourselves one of these famous double helices. These 2 long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. Thankfully, when it comes to figuring out what part goes where, all you have to do is remember that if one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases,
Okay,
or the base sequence.
Let's get that, because that's a common question in biochemistry, and class. Let's go back to here. So you have your sugar phosphate backbone, which is like the rungs of the ladder, you know, the support. Then in the middle, you have your nitrogenous bases. Now, it could be ATGC on either side, but no matter what is on the 5' the 3' side, which is this side, right? It goes, no matter whatever A you have here has to pair with a T, okay? So adenine always goes with thymine with 2 hydrogen bonds. And the guanine, the G, always has to go with a C. So we're gonna see in a moment how that forms a code, and just with those 4 base pairs, you can make quite a complex code. So remember, there's adenine and thymine, AT, and that's 2 hydrogen bonds, and then GC, guanine, and cytosine, that's 3 hydrogen bonds, and that's a classic question, biochemistry question for future. For your future nurses, for your NCLEX class, or any of you going to medical school for MCAT, they always talk about what is the structure of DNA, and you will say sugar phosphate, right, which is here, and we have that 5 carbon sugar ribose, and the rungs of the ladder are these nitrogenous bases connected with the hydrogen bonds. And again, adenine and thymine, AT, is always 2 hydrogen bonds, and then GC is always 3 hydrogen bonds. Okay?
Bond, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCCATG means something completely different as a base sequence than, say, TTs.
Okay. Now we could see a code. Let's go back here. So if we have a code going AGGTCCATG on one side, the other side should match. So we already know that A always matches with T, G always matches with C, A and T always go together, so this would be matched to an A, and the C always goes with a G. So do you see how it matches? And if we could do that, let's pause this and do this as a nice nice exercise. Let's see how we can do this. Control Shift. Maybe I can take a picture of this.
G means something completely different as a base sequence.
Let me take a, because this is also a lovely question on a lot of exams. So let me take a picture of that. And then we have that picture here. Now, let's put it in a Microsoft Word. And then I could show you. I love how Microsoft Word has to verify that you paid. Yes, I did pay. Maybe late, but I did pay. All right, here you go. Put this in here. So remember, we already know that the base pairs, it forms a code, and we already know that all the A's always goes with the T's, and all the T's always goes with an A. All the G's go with C's, and all the C's go with G's. We already know that from that previous picture that we had. So let's match things up. The A will always go with a T. The G will always go with a C. The T will always go with an A. G's and C's always go together, so that's a G, that's a G, that's a T, that's an A, that's a C. So does everyone see how, like, you always have an AT, GC, GC, AT, since this is a T, always matches with the A on the other side. So let's say, for example, this is a 5' to 3'. It's going this way. This way will be going the opposite way, right? And then there'll be double bonds in between. And the double bonds in between A's and T's, A's and T's, A's and T's, A's and T's, and triple bonds, 3 bonds between the G's and C's and the C's and G's. So it forms a code. And that's where he got that part. like AGG, TCC, ATG matches up to TCC, AGG, TAC, right? That's that other part in the video. I believe you said it way too fast. So you could see here how this is a classic question. They'll give you one strand going from one direction, and I'm just saying as an example, like this way, and then they'll ask you, what's the code going the other way? And it's daunting if you don't take a minute and then map it out and just actually draw it out and write it out. But then when you write it out, it seems very simple. Okay. Does everyone get how this, like if, like for example, I could give you a question on an exam, right? And the question would be, for this code, which of the following would be the proper code? Would it be this, this, you know, and I'll, you know, you go on the line. And of course, this one is the correct answer. But you can go down the line. Now you could sit and try to match each and one, but the easiest way to do it is you take the code, you write it on a piece of paper, and then you write, then you just figure out its matching part of the code on the other, and they call it a strand, like a string, and that's the easiest way to do it right here. Okay. So that is a classic question that comes out and will definitely come out on your final, or definitely will come out in other things. Where am I going now? What am I doing with this? I was supposed to go to the video. I didn't want this. Where is my video? Ugh, I gotta go back. Oh, here it is. It's embedded.
Say TTCAGTCG. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's one for each of your chromosomes. Put all 46 molecules together, and we're talking about roughly 6 billion base pairs in every cell. This is the longest book that I've ever read. It's about 1,000 pages long. If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Pop quiz. Let's test your skills using a very short strand of DNA. I'll give you one base sequence. You give me the base sequence that appears on the other strand. Okay, here goes. So we got a 5' AGGT.
Okay. Let's take a moment. This is a classic question. You have a 5' to 3' AGGTCCG. So how would you answer this question? Let's take a moment, and I'm going to screenshot this. And then let's go to Microsoft Word. So let's say we're in an exam, and we get this. Okay. So the first thing I'm going to do is I'm not going to worry about the direction yet, right? But heck, I'll even put the direction. I know if this is going from 5' to 3', the matching strand has to be going the opposite direction. So it has to be going from where? 3', and then all the way over here would be 5', right? So then now all I do is match up the letters. A always goes with T, right? G always goes with C. G always goes with C. We already know that. T always goes with A. C, G, and G, C. And then it goes, of course, if I started at 3', I'm going to 5'. Do you see how, like, if you break things down and you make things slow and simple? And that's the misnomer about medicine. Everyone thinks that medicine or science has to go fast. No, it has to go smooth. You want to do things right the first time. And imagine if you had like 4 or 5 of this, it gets confusing. But if you sit and write it down and know what we already know, I know that one strand of DNA goes one way, 5' to 3', and I know another strand of DNA the matching strand has to go the exact opposite way, 3' to 5'. And then I just match up, the letters, and now I have my answer. And then I just look at A, B, C, D, E, right? And then match up the answers. Also, I will also be doing a primer, or if you have an exam coming up, give me a call, because another thing I tutor is test taking strategies. Because one of the other things that I always mention in other classes and in new student orientation is that, you know how people keep on saying that, oh, I'm a bad test taker. And I'll have this this thing that I wrote. I'll put this in the notes for you guys, so you could practice this at home and look at it at home, and also along with your video. But a lot of students always say, oh, I'm a bad test taker, that's why I didn't do well in the SATs. No, you're a bad preparer. You did not properly prepare. And there's a lot of nurses, almost 30% of the nurse candidates fail their T's at least once. And each and every one that I spoke to who failed it, they did not properly prepare. And I asked them, how many practice tests did you do? And then I get, practice test? And I go, of course. Remember, like a prize fighter, if you know you're going to have a multiple choice examination, shouldn't you practice multiple choice examinations all day? Right? And so you can get used to it. A prize fighter, no matter how good you punch that bag, or no matter how good you watch the video of your opponent, if you do not get in the ring daily, you're gonna get beat up. I go, I know from experience. I spent half my youth in a ring, and all the fights that went well, all the tests that went well because I prepared. And everyone knows what not preparing for a test or studying the wrong material for a test feels like. It feels like defeat, doesn't it? It feels like, I'm stupid. Oh my God, am I the stupidest person in the world? No, you just went into a gunfight with a knife, and you will lose and you will suffer. But the people, like, you also remember the time where the test seemed so much easier because you properly prepared. And part of that preparation is also diet. How many of you eat like a Danish full of sugar and have a cup of coffee before an exam? Wrong. Because that's a good way to ramp up your metabolism. It's a good way to, and what happens when you're nervous? Coffee, caffeine, when you're more nervous, can you remember things? You can't. And then it gets expanded. So there's also a diet that you should, and a routine you should follow, so that when you take the exam, it feels like nothing. My daughter prepared for her NCLEX, I think 14 months. It's a long time. It's like 14, 15 months. She went through 2 booklets and 6 whole NCLEX examinations, as in real. She sat down and stayed for the duration of an entire exam. She finished her exam early, and when she met me out in the parking lot, and she looked at me like, all right, can we go to the mall now? And I'm like, you don't feel nervous? You don't feel sick or anything like that? She goes, no, it just, and she said something that I always say, it's just another day. She scored a 98 percentile.
Wow.
Yeah. But you know what's funny? When she went to go looking for jobs, do you think they care? No, just pass. What they really care about is how good are you in the ward? And that's what I was trying to tell her. She was like, no, my scores and, my grades in school. And I go, baby girl, they don't care about that. They go, did you graduate from an accredited college? She did. Did you have a passing NCLEX score? Okay, fine. The majority of her interview, her interview was over an hour, was about how well do you work with people? Have you had occasions where the patient spit on you, right? Got you in trouble, cursed you out, was either racist or sexist or misogynistic to you, and how'd you handle it? They don't care about your grades, because grades are just one part of the equation. What makes a great medical professional is the demeanor at which you utilize all this knowledge, right? So yes, grades are important. Yes, tests are important. But because that combined with what you're capable of doing in the outside world, that's the important. And this, why are we studying all this? So when you look at a genetic problem, you now totally understand, like, oh my God, I know it's from the code. There's something in your code that's messing you up. Right? And like SIDS, I think was it this class that had a discussion about SIDS?
No, I think that was
No.
no, not that one.
Or medical terminology. I remember it was in one of my classes. But when you look at it, SIDS, it goes, yes, it's a mystery. We still don't know what it means. But now you see the code is very complex. Don't you think if the code gets messed up, your body will say stop? Because they know if something's messed up in the first part of the code, don't you think the rest of the code is going to be messed up?
Well, is that similar to what we were learning about the mitochondria, about how both have to work properly in order to function in the body properly?
Yeah.
The mitochondria, which is a small part but integral part of the cell, doesn't work. You see, it creates a whole bunch of diseases.
Right.
A whole bunch of problems. Now let's talk about something more integral, genetic diseases. You could now see exactly that the code is very, very delicate. And this should amaze you. This should amaze life. I believe, and I'm going off a little bit of a tangent here, I believe if everyone understood how complex and how beautiful and how much of a, for lack of a better term, I don't want to use a religious term, but how much of a miracle just you breathing is. I truly believe people will stop doing the things that we're doing to each other.
Let me ask you.
Fully know and understand how precious we all are.
Right. So let me ask you a question,
Yeah.
Dr. Garais. So would you say that our code, since it's all broken down into letters and all that fun stuff, would you say it's similar to when a say like a tech person, somebody who creates websites, when you're looking at CSS and HTML code,
it's code.
Looking at all that, and you have a page full or like a couple Word documents full of all this code, when you mess up one, the whole thing is
the whole thing falls apart.
Okay, so that's what basically your body.
That's why we as clinicians now fully understand how delicate life is, and also how robust it is, because you could see like, oh, now that I know all these little tiny things can go fall apart, you should wake up every day amazed that you're still breathing. But the system we're going to talk about in a minute has a fail-safe. When things go wrong, it corrects itself. And this should amaze you. It's constantly correcting itself, right?
Okay.
And it's constantly and it's constantly building. And remember I mentioned in last week's lecture, DNA also, now we know for a fact, for NIH did some wonderful paper about 2 years ago, and the American Psychiatric Association confirmed it. About 40% of our DNA structure houses behavior. So there are people who have a propensity to do good things. There are also people who have a propensity to do bad things.
Well, since
but
DNA
again,
is in charge of our chemical makeup physically, wouldn't it also go into know that it is in charge of our emotional
in charge of everything? But now when you know and understand that, right, then you can have then the other half, which is behavior modification, right? Like for example, I know I have a temper, right? And I went into the military and it made it worse. So I was smart enough to go, maybe this is not a good career for me, right? So I went into chemistry because I thought, oh, being in a lab, it was nice, and I always wanted to know about all these things. So, but I have a lot of friends who stayed in the military, got divorced 3 times. They beat their kids, they beat their wife, because what are they trained to do? They already have a propensity towards violence. So they're trained to do what? More violence. And it made things worse. Then it brought on alcoholism. Then it brought on these other things, right? So you can see how it goes psychiatry, it goes and anatomy and physiology and brain chemistry, all of this relate to each other. And the more we know about all of these, we can now mess with it and now modify it. So, and like I said, if more people knew and understood how this all works, because I don't believe people would be doing what they would be doing to each other.
Well, I mean, even then, once we figure out what the core issue is, say, with one individual, at that point, when you say we can change things, that's when potentially some medication can intervene something and correct some kind of chemical imbalance or
Yeah,
something.
and also I am also a proponent, even though my mother's a psychiatrist and she's been, touting all this medical stuff, medicine, drugs. I am not a proponent of that. I'm internal medicine, but I don't like drugs. It
Mhm.
has to be a balance. There
Right.
has to be behavioral modification. Like for years, I didn't want to go to therapy.
Hmm.
I'm like, oh, I don't want some goofy guy with a non-MD telling me what to do. But again, with that modification, with somebody else other than your family, other than your friends, telling you who you are and
Hmm.
and giving you techniques on how to change your code, that's how it works. And it
Right.
works. And
They help you,
over
right?
the
'Cause-
years, I'm not that road rage guy. I'm
Mhm.
not I'm not the typical angry Marine anymore.
Right,
Right.
because
But
they
it
help
took a
you
long time.
talk and work out all the issues and give you tools to work with the issues in a way, I guess, you
Yeah,
know?
so you could see here how how your basic sciences like anatomy and physiology, biochemistry, chemistry, microbiology, all of this gives you like all the secrets, not
Mhm.
secrets, but all the little reasonings, underlying things on how we exist as a human being, right? So
Mhm.
now you know that there's a code. Now let's see now what does this code now mean for us. Now he already explained that now it's the code, and now we know it's the code for everything. It's the code for why I drink too much or why I don't drink at all. It's the code for why I have glasses and why other people don't have glasses. So there's a code. So what does this code actually then translate to? So let's get to that part.
DG to 3 prime, and time's up. The answer is 3 prime TCC AGGC 5 prime. See how that works? It's not super complicated. Since each nitrogenous base only has one counterpart, you can use one base sequence to predict what its matching sequence is going to look like. So, could I make the same base sequence with a strand of that other nucleic acid, RNA? No, you could not. RNA is certainly similar to its cousin DNA. It has a sugar phosphate backbone with nucleotide bases attached to it, but there are 3 major differences. 1, RNA is a single-stranded molecule, no double helix here. 2, the sugar in RNA is ribose, which has 1 more oxygen atom than deoxyribose, hence the whole starting with an R instead of a D thing. And finally, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the
All right,
4th nucleotide is the base.
let's look at why RNA is like the messenger. Now, remember DNA was double-sided,
Mhm.
okay? So this thing's only single-sided. So it's going to take a copy of one of the strands of the DNA and then translate it over. And the only difference with RNA, well, not only, but the main difference of RNA and DNA is, of course, RNA is ribose with an extra oxygen, so they call it a ribonucleic acid. But this is the real thing on exams, the uracil. So you don't have a T, you have a U. Okay, and the classic question that they always have is they'll have one strand of DNA. They'll ask, what is the matching strand for RNA? And remember what RNA is. It's going to take the DNA code and it's going to translate it. It's going to messenger it to our protein factory, which is the rough endoplasmic reticulum, and it's going to make a protein. And we're going to show you that in a minute. But the main thing about RNA is it doesn't have thymine, it doesn't have a T, it has a U, okay?
So it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the replication of DNA. But first, biographies. Yes, plural this week.
All right, I'm going to skip over this part because this is the part of your discussion that they talk about credit where credit is due and scientific documentation, and how many times history is written by the victors. History doesn't necessarily mean it's true, right? And the classic example is they've always said in history books, even up to when I was in college, that Watson and Crick discovered the DNA helix and won the Nobel Prize for it. But it isn't true. They plagiarized off of somebody, and that person died. And we're going to talk about that lovely lady in this next part of the video. We're at 6 minutes and 24 seconds. So jump to 6 minutes and 24 seconds when you work on your discussion, and then you could do, of course, other research to validate. Don't take this guy's word for it, but to validate, is Watson and Crick the true originators or the true discoverers of the DNA helix. So I'm going to skip over that part. And also, if some of you are looking at that part, you'll be thinking like, oh, women again being, being exploited or whatnot. Just know that for the ladies in the audience, you are now in healthcare. Healthcare is female-centric, if some of you haven't noticed yet. When I was in training, all but one of my attendings were male. Because in medicine, no one gives a darn anymore. Male, female, or whatever pronoun you want to pick. Even in my mother's generation in the 70s, my mother graduated neurology from Yeshiva University in New York City. Yeshiva University used to be all Jewish males, right? If you look at her graduation picture, it's 20 dudes and there's one little lady. And my mother was chief resident.
Wow.
Why? Because she's no smarter than anybody else. But remember, the hardest working tool in the shed. My mother is the eldest of four. All four children went to graduate school, all four children, summa and magna cum laude, except my mom. But my mom was the only one with the MD, right? Why? Not because so if you're not one of these people who are like instant geniuses like my aunts and my uncles, right? My uncle was a raging alcoholic and went through graduate school like it was a joke.
Wow.
He wouldn't read, he wouldn't do anything, right? But me personally, I'm not one of those people either. I totally believe in hard work beats anything all the time.
Yes,
The
I agree.
and the last one to go to sleep beats everyone every time. And actually, isn't that the kind of medical professional you really want? You don't want a smarty pants, right? You want somebody who cares and who stays with you. Hence the term, if you guys know the term, resident, right?
Yes.
Resident means what? You live there. That's why I find it very funny when the nursing staff is like, I've been here 17 hours. I tell them, I've been here 17 days. I don't understand what you're complaining about. But the hardcore nurses, they don't complain. They're there day in, day out, 20-hour shifts, six days a week. And my daughter's one of them. And they do the job because, number one, they love science. They want to know the answer. And number two, they want, oh, thank you, big boy. Son brought me some food. Right? The advantages of online education. You can get food while you're teaching. Right? It goes, and it's the same thing. And also to mention my son. I always want to mention my son. He's 14 years old. He's on the autism spectrum. I took him out of school because they always said, oh, well, this is all he's good at. BS. Right?
Yeah.
He now did, in this last year, he did 95,000 questions online.
Wow.
95,000. I don't think I could do 95,000, but he did it, and he works every day. And the last IEP told me, oh, he can't sit in a room by himself for 5 minutes, right? So I could sit and listen to someone else's data, or I can do what? Create my own data. Now, I can say to you, oh, it was easy. No, it wasn't. The last 4 years have been really, really hard, and really, really backbreaking, and it was really hard for him, and it still is. But because he has to wake up he goes very early, and he goes to sleep late because he will not go to sleep until the work is done. And it's 7 days a week. Right? There's no weekends for us. So if you guys are getting used to weekends, please do not. You're in healthcare. It's 7 days a week, and that's what makes us also special. People don't look up to us because we're any smarter, or they definitely don't look up to us because we're making money. They look up to us because we are there at the worst moments of their lives all the time.
Yes.
Right?
True.
And that is why we're there. And that's a pride that's hard to get rid of. So when this stuff gets hard and esoteric, especially a chapter like this, where it's kind of weird, think of that for a moment, that this thing you're learning will help me fix somebody, or at least decrease the level of pain. I was so happy with the nursing staff yesterday at Patient First. The MDs really wouldn't have cared less about my pain yesterday. And I was being, of course, a typical male patient. I was being a big baby, but they gave me some nice happy juice, and that made things so much better. I was higher than a kite yesterday, but legally, right? But my God, I was in so much pain. But hey, it is what it is, and I appreciate the staff. And I just find it sad that the MDs didn't appreciate me as much as the RNs and even the MAs who were in the room. And I appreciate that, because I used to be. So don't look at yourselves like low person on the totem pole. When I was in MA and when I was in EMS, I used to be like that, like, oh, I'm just a EMT, no one cares, you know. But you know what? You're there when people aren't. And the MD, yes, they're important, but everybody else on the team is equally important. And without that support, the MD can't do their job.
Exactly. I feel like in this kind of field, that's why I'm going in. I'm not going in to get any kind of glory because I'm smart or.
Yeah, you're not gonna get any.
I just want to be there for somebody who's not feeling well. I just want to be there for that person that doesn't know what's going on, and I want to help them find answers for what's going on and help them treat.
And it's just cool. And they won't say thank you. They'll actually spit on you.
Yep.
They'll actually like, why didn't you do this to me yesterday? And you're like, oh, okay, thank you, have a nice day. And then
I worked in retail. I've already dealt with that. So
if you worked in retail, then that's perfect. Like if you worked in any job like retail, or for a year in high school, I worked on a complaint line for In Fashion magazine. Oh my Lord, people would curse me out for the littlest things. I didn't get my volume 6. I hate you, mother jumper. And you're like, I'm gonna bring my son over there, beat the crap out of you. I'm like, what the hell? And I'm like, sweetie, I will get you your damn magazine. Why are you threatening me?
I worked at Barnes & Noble, so they're like, well, I can get this cheaper on Amazon. I'm like, okay, then go ahead.
And then they'll complain, aren't you supposed to keep me as a customer? What kind of employee are you? What's your name? Who's your supervisor? And you're like, oh, Jesus. 8 hours of this. 8 hours. And you're like, and after the day, you just want to kill yourself. You're like, why am I doing this? And then you remind yourself, I want to do this so I can go to school and then do what I really want to do. And then when you get to do what you really want to do, it's the same thing. Yep. It's just on another level. And you're like, right?
Well, you know, at least being in retail or some kind of waitressing or something like that, working
customer
those
service job. Job.
They give you a thick skin. They teach you.
Yeah, but you guys know, all of you guys know, work in medical or not, you know that the hardest part of the job isn't the job, it's the people that you come across, and even some of your colleagues. I used to think in medical school, like before I got into medical school, I was thinking, oh, after graduate school, I'm going to be with other people who are like me. No, because these people were jerks in college. They're going to be super jerks in graduate school. Because now, doctors, a lot of them are arrogant, because think about it. If you're the one who survived out of all these other people who really, really smart people who didn't survive, it kind of gets to your head. And I'll admit it, for a little while, if you guys met me as a resident, you'd be like, oh my God, what an arrogant dude. I don't want to work with that guy. Actually, when I go back to Lincoln Hospital in New York, I teach there every year in a symposium. But this year I didn't due to COVID. The nurses always ask me the same thing. Dr. Garais, you coming back? As if like, oh, we don't want you back. I'm like, no, I'm not coming back. What the hell's wrong with you? Right? But they look at me like, oh, that's good. I'm like, oh, I didn't want to come back either. So what do you? And some of the junior nurses now are head of staff, and some of the junior doctors I used to work with now are chiefs of their own staff. And I always get that question, because I used to be a very typical totalitarian, hardcore leader. My way or the highway. If you don't agree with me, I will ruin you. That kind of person. But then when 20 years ago, when I got into teaching, I saw the world very, very differently. And then little by little, you change from that. And by the way, if you're that kind of person, it may work for your kids, but it will definitely not work for your team. You can't be that Hitler type, Mussolini type person. It won't work. I mean, yeah, it will work for a little while, but your reputation will follow you, and a lot of people won't want to work with you. To be even-headed and even-keeled is the best policy, especially when you get angry. And like I mentioned earlier, I shared with you guys, when I was younger, I was a hothead, so that's never a good thing. Even though you're smart and you know stuff, being a hothead and being emotional is not. I mean, it's cool for those movies you watch regarding, if you guys, some of you guys like, doctor movies or doctor television shows, but in real life, the best clinicians are the ones who are like, something awful is happening. They go, okay, guys, let's calm down. Let's look at this. They're the ones who keep everybody calm, right? But when I was a resident, I was not one of those people. Okay, so
it turns out that our cells are extremely good at this. Our cells can create the equivalent of every cell in your body has a copy of the same DNA.
Now, remember, we always have to keep on our goals. So how does this DNA replicate and double up and transfer, grow? So that's this part.
Started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. So, how is a teenage boy like the enzyme helicase? They both want to unzip your genes. Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs.
Okay. How do I know this is DNA? Two things. Once, it's double-sided, right? We see that. And then, do we see any uracil or U? No, I see a bunch of T's. So this enzyme, helicase, and we already know what enzymes do. They break down things, right? How do I know it's a enzyme? ASE, right? So helicase, in order for this to replicate, helicase has to unzipper the two sides, and that's step one.
The splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand, as I call it, another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt to deal with. These unwound sections can now be used as templates to create two complementary DNA strands, but remember, the two strands go in opposite directions in terms of their chemical structure, which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading, good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it can do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, you can thank the enzyme RNA primase. The leading
okay. Now, all that stuff that he said, now when you unzip a DNA, now you have two copies, but they're going in different directions. Now, the leading strand is what he called the good guy strand, is easy because it goes from 5' to 3'. So it's going in this direction, right? Now, you have a primer, and what does a primer do? Prime means one, or starting. That has to start it, and then it can make copies all the way down. Now, for the leading strand, it's easy because you're going from 5' to 3'. Now, with the lagging strand, it's difficult because it's going backwards. It's going this way, right? And DNA polymerase, right? Poly means many, right? You have a DNA, you have a primase first, and then you have a polymerase that will go down here. The polymerase can only go in one direction. So it's going to be weird on how we're going to do the lagging strand. So let's watch the rest of this video, and then we're going to talk about how your body does the lagging strand. So this is step 2. Step 1 was helicase. Step 2 was primase. And then you have a DNA polymerase that will go down and make copies of this chain. And you could see it's forming another DNA strand.
Needs this RNA primer once at the very beginning. Then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging or scumbag strand is, well, it's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5' 3' direction, and the lagging strand is 3' 5'. So DNA polymerase can only add new nucleotides to the free 3' end of a primer. So maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here that awesome little enzyme, RNA primase, does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point, to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. They're called Okazaki fragments, after the couple of married scientists who discovered this step in the process.
Okay, so you can now see, let's look at the other picture previously. The way the lagging strand works is, remember the primase? It goes somewhere here in the middle, and then it works 4 or 5 pairs down, and then the DNA poly follows it. So it makes the lagging strand in pieces. So at first it'll do like this piece, and then piece, and then that piece. And those pieces are called Okazaki fragments, right? So your lagging strand has to be, it goes in the other direction. The leading strand is easy. You have DNA primase, that's easy. And then polymerase goes all the way straight down, and then you make a copy. But the lagging strand is difficult because it's going in the opposite direction. So it'll do this piece first, then that piece, and then that piece. And you could see that there's a potential for errors. But let me just clue you in. DNA poly, or DNA polymerase, has a nice, it's kind of like a proofreading function. So it makes sure that, is this correct? AT, GC, GC, CG, that's good. AT, ATTA. As it's going down, there's a piece of DNA polymerase that proofreads this. All of this should amaze you because this is happening right now 1,000,000 times over, and you're not dead. And your cells keep on propagating the proper code. But you can also see, let's say if there's a piece of the code here that says you have cancer, what do you think it'll keep on doing? It'll keep on telling the rest of the cells, hey, it's time to make some cancer. And that's a problem. And that's why we're really working on genetics. Like a lot of laypersons say, oh, we're working on genetics. It's going to be, we're going to do some Hitler stuff and make everyone like blonde hair, blue eyed. No, we wouldn't care less about blonde hair, blue eyed. Medical science cares more about, we know that this is the true source of disease here, somewhere in here, right? And if I can fix this, then what could we do? We could decrease the propensity towards heart disease, decrease the propensity towards cancer. Now, of course, remember you have, if you recall your biology, you have this, which is your DNA, which is the blueprint. You also have, the term is nature versus nurture. So our nature is our genetics, but there's also nurturing. There's something called the 11-year rule. Like, yes, Tiger Woods is a great golfer, but we didn't see the 11 years of him training, right? Since he was 3, right? So yes, he had genetics to be a great athlete, but if his highly abusive father did not put a club in a 3-year-old's hand and then tell that kid, you will not let go of that club and it will make you a bazillionaire, right? So it needs 2 things. And you can see that, and I'll tell you, there were so many students in medical school who were crazy smart, and a whole bunch of them failed. Because why? There's also nurture. There's also practices outside your DNA. Right? My DNA says that I'm okay at school, not great, but the discipline that I had from teachers and my parents helped me be a physician because it enhance this DNA. My sister is a genius. We found that out in second grade when she got taught chess in second grade, and she beat her teacher, and then beat him again in front of the whole school.
Wow.
Yeah. So they tested me because they tested her and they found her IQ to be 142. So she's a genius, right?
Oh my God.
Yeah. So I'm going, oh, so the school's thinking, oh, there's got to be a reason why the other one's stupid. So well, I got left back in second grade. So they were like, oh, why is this one not getting it? Because they had the idea, maybe he's bored and he's secretly a genius. Nope. Guess what my IQ is? 96. Completely average. Actually, it's a little bit on the low average scale. So how did I become the doctor and my sister didn't? Right? But my sister also did some crazy stuff. She went into nursing. She left nursing for this little thing called IT. Because when she was a nurse, she was noticing like, this was in 95, 94, when people didn't have real internet or anything. And she goes, I think this information technology is really going to blow up. Now she's an executive, and now she runs an IT department in California.
All the way down
in California.
Lagging or
but you could see
Strand.
what I've always been saying: if you do not enhance your genetics with practice, right? Like Tiger Woods, like Michael Jordan, like the Michael Jordans of this world, right?
Mhm.
All the brains and all the DNA will be for nothing if it doesn't get practiced. And also, you could have moderate DNA, or you could have, like in the case of my son, damaged. But you could see he does way more work. He reads at a collegiate level at the age of 14, right? They told me he would never be able to, that he would have difficulty like counting money and things like that. No, because what did we do? We kept on working at it, and we're diligent, and it's 7 days a week. I don't think in the last 8 years he's ever had a day off, and I don't want to give him a day off because he's used to it now. And it's the same thing with you guys. You guys only got a day off. So now, what happens when you go to the ward? You'll be the better clinician, the better nurse.
Mm-hmm.
So now you can see like how all of this plays together, and also how complex all of this is. I'm gonna pause this video now because, and also how now we can look at DNA structure and and reading the code and copying the code, how important this all is to your actual functioning. And that's also why on your, when we ask for your past medical history and we ask you for your genetic history, like, hey, if you've ever been to a doctor, they always ask, hey, are your parents alive? Do they have any illnesses? Because now I get a glimpse of your code, right? And then clinically, I can now go figure stuff out. So let's look at a little bit more genetics. And of course, for this week, the case study is the dividing the cell, right? So of course, you read it. It's kind of corny, but this is a way to look at the cell. So do this like the 1 or 2 questions here. There's not that many questions, but read over this. It's, like I said, it's kind of corny and I don't want to go over it, but it's also a nice little review of how cells separate. And there's also another video that I want to go over that is also important and that will match to this video right here. So there's only 1 or 2 cases for the case study for this week. And of course, you can even break down this video for your concept map. But this video is the majority of the lesson for today. Now, what is this thing? I never used to click on this. Now, let me click on it again. Oh yeah, nice to know, but if you want to look through it, DNA extraction and things of that. And of course, like I said, we're not doing the cheek cell DNA extraction, but if you look, there's nice notes in here and the stuff that we were talking about previously. Okay. So let's go to that video regarding, and that's in the task. So this video here is in lesson 4. But remember, the only thing you're handing in lesson 4 are the case questions here in this application assignment. So let's go to this. And then it's in task 4, I believe, the video on mitosis versus meiosis. So yes, click and go through this video, but you know me, I love charts. I love like, and there's this chart that I almost always use is this one. And this is a nice little comparison, but I like this comparison right here on what mitosis and meiosis is. Let's see if I can get this bigger. Oh, there you go. Wunderbar. Okay, mitosis and meiosis. Now, all of them have to go through some sort of, now we know exactly how these chromosomes are formed, and we know we have 46 of them. And if you have 46 of them, you have 2n, right, and lowercase n, number of genetic material. That means you're a complete human being, right? So in order, and we also now know how DNA gets opened up and how we unzip it and how we copy it, right? And let's go over mitosis first. Mitosis is for somatic cells. And what does that mean? Soma means body. So if I have a cheek cell or a skin cell, right, the cell should be the same exact copy, right? Like I don't want the cheek cell to look like an eyeball cell. I want a cheek cell to look like a cheek cell every time. So in mitosis, I'm creating somatic cells. Therefore, I start off with 2n DNA or 46 chromosomes, and then I will end up with what? I should end up with the same amount of genetic material, but split up, okay? So I start off with 2n, and then I end up with 2n, okay?
Mm-hmm.
So because the cheek cell has to look like a complete cheek cell. It can't look like only half. So mitosis goes through prophase, metaphase, anaphase, telophase. This prophase, metaphase, anaphase, telophase, which is, if you read your discussion case, they go over that in some corny fashion, but read it anyway, right? And the DNA doubles up, and then it splits, and then the daughter cells should all be the same, 2n, right? So 46 chromosomes here, 46 chromosomes here, okay? So the daughter cells should look exactly like the parent cells, and that's mitosis. It only goes through one set of duplication of DNA and then one set of splitting, okay? And so we start off with 2n, we end at 2n, which is a classic question regarding mitosis. And it makes sense because I want my skin cell to look like a skin cell. Now, that's mitosis. I don't know who named these things. It's named so close. But when you have things that are so close to each other, memorize one like your life depended on it, and the other one is simply the other one. Now, meiosis, they go through not one, but you could see it goes through meiosis 1 and meiosis 2. So it splits twice. So it takes this genetic material 2n, we double it up, and then we split it, and then split it again. So if I split up the genetic material twice, I'm going to end up with 1n, or 23 chromosomes. So the daughter cells will be germ cells, and we call them germ cells, not germ as in, you know, antibiotics germs, but germ cell as in sperm or ova. Because remember, you get 23 from mommy, which is a single ova, and 23 from daddy, which is a single sperm, right? So that is where meiosis is utilized. It's utilized in germ cell layers, which is human reproduction. So what kind of question could I ask? I could ask, what do you start off with in meiosis? Well, 2n. How many divisions does meiosis go through? 2, right? And look, it has an I and an I for some reason. I see that as 2, right? Like the Roman numeral 2. And then, so we start off with 2n, and then I end up with what? 1N. And the mitosis only goes through once. I start off with 2N, and I end up with 2N. And 2N means 46 chromosomes, complete. 1N is only 23 chromosomes. It means it's a germ cell. It means it's either a sperm or an egg. And that's what meiosis is. Okay? So this is a nice chart. You could pause it and or even just Google it and you can look at it. It's like the second or third one in the in the search. Just right here. And also the Khan Academy goes through something similar as well. Okay? So, do we know DNA replication? Yes. Do you know mitosis versus meiosis? Yes. Do you know the structure and the function of DNA and what does it actually do? Yep, we went over that. But we didn't quite go over protein synthesis. We'll continue that next week. That's for next week. But now you know cell growth and division, cellular differentiation. You knew from last lecture, right? And nucleus and DNA replication, which we already now went in full detail. So does anyone have any questions?
No, I think I'm all set.
You guys are all good. So if that's all good, I will see you guys next week. And what is due today? Task 3, discussion 3, lesson 3. So please submit if you haven't done so already. And I'll grade it later on tonight.
All righty. And are you gonna keep us informed about when we're gonna be having class times and all that fun stuff?
I'm gonna be going through labs. I'll be putting that in the announcements later this afternoon because it just got approved. So, but again, if you can't make it, please give me a call because I can put you in with another class, or we also have Saturday mornings. Okay.
Okay. All right. Thank you.
As like because I know you guys are working and stuff like that. Okay.
Thank you so much.
I'll talk to you guys soon. Have a good one.
You too. Thank you. Bye-bye.
Thank you.